Hi everyone, my name is Anastasia Unitt and I'm a DPhil student within the Maiden Lab group, and I'm going to be discussing a case study of how to use the PubMLST database to study protein sequences, which is what I did using the bacterium Neisseria gonorrhoeae. Now, STIs and protein sequences. While nucleotide data can tell us a lot about an organism, protein sequence data represents a step closer to phenotype. With the vast amounts of whole genome sequence data now available, it is quite simple to gain insight into protein sequence by translating this nucleotide data. For example, in my project, I've been looking at the protein content of Neisseria gonorrhoeae outer membrane vesicles, or OMVs. These structures are implicated in pathogenesis and are of interest for use in vaccines. My work built on the foundations of a methodology that was used by my colleague Dr. Charlene Rodriguez to form a typing method based on the deduced protein sequence of the contents of the bacteria vaccine. So the PubMLST database. Databases tend to focus on either nucleotide sequences or peptide sequences and often lose the contextual metadata of the isolate from which those sequences were extracted, such as the year of isolation, location, and MIC data. PubMLST, however, is really flexible and allows you to examine all these different aspects in an integrated manner. Changes to the database are easily altered or updated by curators, and anyone can become a curator, which means that you can add and edit data within the PubMLST database yourself. Now, PubMLST hosts whole genome sequence data for more than 100 species, and that includes many prominent causes of sexually transmitted disease, such as Neisseria gonorrhoeae, Chlamydia trachomatis, and Treponema pallidum. For more information on PubMLST, you can see the website itself or the Maiden Lab YouTube channel for recordings of our previous forums, which go into it in more depth. So the pros and cons of analysing peptides through whole genome sequence data. So in terms of the positive, there's lots of data already available in databases through whole genome sequencing, which means that you can do population level studies relatively quickly, instead of only focusing on protein sequence data from a limited number of isolates. Another benefit is that compared to studying nucleotide sequences, you're less influenced by synonymous changes. You're just that one step closer to phenotype. The downsides is that when working from whole genome sequence data, and therefore nucleotide sequence data, you only have the direct translation, the deduced peptide sequence, protein sequence, to analyze, which means you don't have much insight into post-translational modifications or the level of expression. Additionally, unless your protein has a really specialized and unique function, it's probably going to be expressed in more contexts than the specific one you're interested in. For example, I was interested in my proteins in the context of the outer membrane vesicle. However, all of those proteins are also present generally in the outer membrane of these bacteria as well. So that was something I needed to consider in my analysis. I just wanted to briefly mention some other applications that you could use this kind of protein analysis for. So you might want to look at vaccine components, as Charlene did, or surface epitopes. You might want to look at a protein network specifically, or investigate protein sequence evolution through time. You could also use it to identify representative proteins or proteins of interest. For example, if you wanted to know the most common protein variant for your species, or for a particular location in the globe for your species, you could do that through this methodology. So for the next bit of the presentation, I'm going to describe how I use PubMLST to analyze deduced protein sequences for the gonococcus. So if you want to analyze a group of proteins through multi-locus sequence typing, which is what I wanted to do, your first step is to create a scheme. Now, a scheme describes a collection of loci or proteins from which allelic profiles are defined, such that each unique combination of alleles across the loci in the scheme represents a unique sequence type. This screenshot from PubMLST shows isolate that has been annotated with my OMV scheme, which was of 26 proteins, and because it has a complete scheme annotated, it has been assigned an OMV type, which is 3259. And on the left, you can also see that lots of other schemes have also been defined, which is useful if you want to analyze associations. For example, there are schemes which catalog genes implicated in AMR. In the case of schemes made up of proteins, it is the unique combination of protein sequence variants that we'll be identifying for RST. And multi-locus sequence typing represents a really convenient methodology for capturing and examining bacterial genetic or proteomic diversity. So you want to make a scheme. The first step is to decide what proteins to include in that scheme. And this is a really good point at which to contact curators for advice on which genes are likely to cause you some problems. This plays into your considerations of timescale, which will dictate how many genes you'll have time to translate into proteins and then analyze. but also which ones you're going to use, because some are easier to annotate than others. For example, in my scheme, I had to remove some opacity proteins, which are found in OMVs, because they're known to be really difficult to annotate. Now, moving on to how PubMLST stores alleles. So each gene defined in PubMLST has a continually growing list of defined unique alleles associated with it. This screenshot, for example, shows allele 144 of the Neisseria phet-A gene in PubMLST. Now, PubMLST nomenclature dictates that genes from Neisseria species, which is what I was working on, of course, are assigned the prefix nice, followed by an arbitrary 4-digit number. phet-A, for example, is named nice 1963. Different species will have slightly different naming conventions. So nucleotide alleles will include alleles that are made up of coding sequence and also alleles that are non-coding, and each are assigned an allele ID number. This allows easy comparison of alleles in terms of their diversity and distribution across the bacterial population. Now, moving on to the methodology for creating your protein sequences, your protein variants. So this is a multi-step process. First, the nucleotide sequences for each locus are annotated across all the isolates you're working with. In many cases, annotation will already be extensive, both through automated tools and through the work of other curators, and you will only need to annotate the more complicated new alleles. Your allele sequences, your nucleotide sequences, are then translated to provide deduced protein variants for each protein. For my project, proteins were assigned the prefix nice-P to indicate that they were distinct from the nucleotide locus. For example, here you can see that nice 1963 or phet-A became nice-P 1963 for its translated protein counterpart. Each new protein variant identified is assigned a unique variant number, just as allele numbers were assigned for the nucleotide sequences. However, the difference is that in the event of a non-coding allele, you assign a variant naught or null, because no protein sequence will be produced from a non-coding allele. Once an isolate has all the proteins in your scheme annotated, it will be assigned a sequence type, which you can then use alongside your scheme for further analysis. So PubMLST has several methods by which you can analyze your data, which includes some handy plugins. So I used PhyloVis to help me cluster my OMV typing profiles and to visualize relationships. But I also use Grapetree, which is really great for visualizing the diversity and associations of your schemes, as I will show you in a moment. Export dataset, meanwhile, allows you to easily pull out your data into a spreadsheet, your list of alleles for each isolate, for example, with your sequence types and the associated metadata of the isolates to which they have been assigned. You can then examine this data in other programs, for example, to apply statistics to it in R, which is what I did. So I won't go into too much detail here, but here's a little bit of my data analysis. So this is a minimum spanning tree drawn in Grapetree based on the 26 proteins that were in my OMV typing scheme. And then the nodes have been colored based on different schemes. So at the top, it's colored by the gonococcal OMV typing cluster that they had been assigned to. The bottom left is colored by core genome grouping, and the bottom right is colored by NG star, which is an AMR typing scheme for the gonococcus. So what I was doing here was trying to quickly visualize to see if I could pick up any associations. And you can see if you look at the different trees, that the different patches of color are kind of correlated across the trees. So this indicated to me that there was an association going on, which I then followed up with statistical analysis, which I won't go into here, but that's the logical next step. Now, another thing that PubMLST has built in that it lets you do very quickly are field breakdowns. So once you have compiled a data set, you can instantly visualize associated metadata from that list of isolates, such as year or country of origin, in quite a few nicely presented ways. So for example, if you wanted to have a map which showed what countries your isolates are from, you can have that. You can have histograms, you can have pie charts, things like that. It's a really easy way of quickly generating a diagram to help you understand your data better, but also to present it. So what I want you to take away from this short talk is how PubMLST provides a flexible collaborative platform that you can use to analyze both nucleotide and deduced peptide sequence data from your species of interest. By building sequence typing schemes, you can begin to quantify diversity and look for association with other pre-existing schemes. PubMLST is maintained by the Maiden Lab and a wider group of curators and collaborators, meaning that there is a whole community to draw from for help with your project. For example, in my case, I built on previous work by Charlene, and with her help, I found it much easier to build my scheme and to troubleshoot curation issues. I strongly recommend reaching out to the curators assigned to your species of interest when you do use the database. And finally, my acknowledgements. Special thanks to Dr. Harrison, my supervisor, and to Dr. Charlie Rodriguez, who was so helpful. And if you want to learn more about my work on OMVs, I'll be presenting it in more detail at the STI and HIV World Congress in July. Thanks very much for listening.